So let me share my screen, and I'm going to move into full screen. Just so please let me know if you see my screen properly.
Yep.
We can see the screen very well. Thank you.
Okay, very well. Okay, so well, a very warm good afternoon or good evening to those of you in Asia who are brave enough to attend a session like this after what must have been a long day, and I hope all of you and your families are safe, especially in the current situation and considering the subject of the day. So the purpose of today's session is to give you a little bit of an overview. It will be almost 33,000 feet considering the time allocated, but on the COVID-19 vaccines, but also their acceptability and what's the outlook going forward, and this is why. Indulge into this joke in the title of this presentation about, are we seeing the light at the end of the tunnel? And if so, what do we call the end of the tunnel? Very briefly, as Salman was saying, well, this is what I'm doing. This is not a joke. This is my actual current scorecard. So very briefly, after many years in the life science industry, both in big pharma in different geographies, and 3 times as CEO of startup companies, and leveraging a background with an academic background as a Doctorate of Pharmacy, a Master in Business Law, and an MBA. I'm running my own advisory practice, which essentially is a shell to host all my different activities. I happen to be, after a stint at the board of that company, I'm now heading international business operations for one of the companies which is involved on the front line of procuring COVID-19 vaccines, and it's a Chinese company called CanSino Bio. Besides this, I'm holding a few board positions in companies. Very quickly, Theradiag in vitro diagnostics, public company in France, Eurocine Vaccines, vaccine company, early stage, public in Sweden. My CB1 is therapeutic cannabis, private in the Netherlands. Vaccitech is a British company, vaccine and cancer immunotherapy, private, but preparing its IPO in the UK, in Oxford. Univercells is a Belgian company, still private, also preparing its IPO in the field of production of biologics. And Adiposs is a Swiss company in the prevention of cachexia in cancer patients and chronic, chronic patient with chronic ailments. And besides this, I do a bit of investment advisory, being the regional partner of a French family office, which I represent in Switzerland, and being the chairman of an Italian family office based in Milan. And yes, believe it or not, I still have a bit of spare time to mentor some of the startups at the Mass Challenge in Switzerland. So that's what I do for a living. Usual disclaimers: in some of the companies I'm working for are public. All the ideas expressed in the presentation are entirely mine. So if you need to sue someone, you sue me, but nobody else. On that note, let me introduce you very briefly to what I was planning on discussing with you is the following. Maybe, starting with a few, quick considerations about vaccinology, because not everyone is aware of this science. Mind you, my first position in the field of vaccines was back in 1986. So I've been seeing a few things in the meantime, including the H1N1 pandemic in 2009. There's a fantastic typo here on, it's actually not Mac 2020, it's actually March 2020. I'll come to this in a minute. We'll be talking about also some of the doubts that float around the timelines of the development of the current coronavirus vaccines, and why this rush development could explain the average level of acceptability of those vaccines, and how could we maximize this in the best interest of public health. And I'll conclude with a few statements about, okay, are we going to be better this time around to draw some lessons from the current pandemic? certainly better than what we've been able to do at the end of the H1N1 pandemic. So let me start with a few introductory statements. The first point that everybody should keep in mind is that the principle of immunization is to trigger a similar or better immunity than the disease without causing any of the risk of the disease. So normally when someone is exposed to a disease to varying degrees, you actually develop an immune response, which is more or less lasting. But the issue is, if you recover, everything is fine, but you might actually die or remain permanently disabled. So the principle of vaccination is really about triggering an immune response and the memory without any of the consequences of the disease. And this is very quickly how a vaccine works. So you see the little syringe that goes through the skin. Of course, you could have a similar graphic with mucosal vaccines, such as intranasal influenza or oral vaccines, for instance. Essentially, it's all about bringing vaccine antigens, which are represented. I don't know if you're. Let me see if I can have a laser pointer here. Here we go. Yeah, you see the little vaccine antigens are presented to the dendritic cells, which role is actually to internalize those antigens and to present them to immunocompetent cells and essentially trigger an immune cascade, which results in the triggering of the B cells and the production of antibodies, but also the triggering of the cascade of the T cells, which are there essentially to neutralize the cells that are infected by the pathogen. Now, very quickly also, what are the ingredients in the formulation of a vaccine? I think it's important to keep in mind because many people are actually worried with what they're being injected with. And of course, I'm sure you've seen plenty of hair-raising stories on the social networks about the formulation of the vaccines. Well, essentially, a vaccine is essentially water. Okay, there's an active ingredient which is usually in very small quantities, which could be, and I'll come to this in a minute. Various types of antigens. You could have adjuvants, which are there essentially to enhance the inflammatory cascade and therefore the immune response. You could have preservatives or stabilizers because, mind you, vaccine active ingredients are typically large molecules that have a nasty tendency to aggregate, to fold or to unfold, and you need to stabilize them so that they actually are in the form that is required to deliver the immune response. And you may have some minuscule traces of substances that are used during the manufacturing process, and that are usually well under the detection threshold of modern analytical methods. Now, let me very briefly introduce.
Yeah, kein Problem.
Sorry, is there a... No. Okay. So very quickly about the vaccine technologies. This chart presents you with the good old ones, so to say, the time-tested ones. And I'm not going to go through all of them, but essentially they use whole or parts of a bacteria or of a virus, more or less modified. The killed whole organism, mind you, is the solution that's being used by two of the current coronavirus vaccines, the one from Sinovac and the one from Sinopharm, which essentially the active ingredients is essentially made of SARS-CoV-2 viruses. And then you've got some modern technologies, including the viral vectors, which is already available for the Ebola vaccine and which is currently the technology, I'll come to this in a minute, used by Johnson & Johnson, by the Sputnik vaccine, by AstraZeneca with the Oxford technology, and by CanSino. You have the nucleic acid vaccines, either DNA or RNA, and the cases in point right now are the Moderna and the Pfizer-BioNTech vaccines. There are some interesting approaches, but they're still experimental with bacterial vectored and antigen-presenting cells being themselves injected. But this is not the case of today's presentation. If we're going to the next slide, let me explain to you very briefly one important notion that is in everybody's mind, and certainly in the mouth of the public health authorities, which is herd immunity. Now, what is herd immunity? How does that work? From left to right, what you see is that if you have a no vaccination or a population which is entirely susceptible because it is not protected by a previous epidemic, essentially one infected person can contaminate the whole population, depending on what is called the R factor. The R factor is how many people is one infected person likely to contaminate. The highest R factor that you can find are with measles, which is around 18, or with varicella, chickenpox. Fortunately, with the coronavirus, we're looking at an R factor which is anywhere between 1 and 3, so it is not that bad, so to say. What happens when you start, having some coverage of the population, either through vaccination or through disease-induced immunity, the propagation of the disease is actually happening more slowly, but it can still happen as soon as you still have some susceptible people in the population. So what you need to reach is a certain threshold of collective immunity, so to say, so that it becomes close to impossible to the disease to spread, except through maybe tiny focal epidemics that you find in specific communities, for instance. What has been assumed is that in the case of the coronavirus, we need to get to 70 to 75% of the population conveniently immunized, and that raises the question of the variants, and I'll come to this in a moment, in order to block the transmission. And as you would assume, other than Israel, there's not one country which is there yet, not even counting the contribution of the disease itself and the contribution of immunity. So we still have a long way to go before we reach the coveted herd immunity and we see a genuine improvement in the situation that we're currently seeing. Now, a little flashback. It's in March 2020 that the WHO declared the coronavirus epidemic a pandemic. It was known that the virus was circulating. I mean, that was a bit fast for me to understand what it was, but so let me first introduce you to the enemy. One of the cornerstones of vaccine science is microbiology. We need to understand the enemy in order to be able to know what we're dealing with. And this is a sketch of what the coronavirus is. Essentially, what you see is those little cones looking like ice cream cones. Are the so-called spike protein. You do have some membrane protein, and I'll come to this in a moment, that they play a role. You got also some nucleocapsid protein, and you got the single-strand RNA, which is in the virus. And that is the beast which is currently crippling the world economy and unfortunately having a too high mortality and morbidity and that mobilizes all the efforts of the scientific community worldwide. Now, it's important to know that in this complex organism, everybody's talking about the spike glycoprotein, okay, which clearly is the target of the neutralizing antibodies. But the antibodies are also targeting. There are doubts as to whether they target the envelope and the membrane protein. They do target also the nucleocapsid protein. And interestingly, whether CD4s or CD8s, the cellular response is actually not only targeting the main structural proteins, so the spike, the envelope, the membrane, and the nucleocapsid, but also a number of other non-structural proteins. Now, this is very, briefly and almost schematically why it is so important to have a balanced immune response where you do have antibodies, especially neutralizing one, but also a robust cellular response so that you have a stronger foundational level of immune protection. Now, when you look at the vaccines, and you have here in this table the front runners, so you have AstraZeneca, of course. I happen to know, just as an aside, in interest of full disclosure, to know very well the technology because the ChAdOx1, which stands for chimpanzee adenovirus Oxford strain 1, is a technology that's been invented by the Jenner Institute in Oxford, and the technology is licensed to AZ only for the coronavirus. And the licensee for all other applications is to Vaccitech, and I happen to be at the board of Vaccitech. Moderna and Pfizer, which is actually BioNTech technology, are both, RNA vaccines. J&J is working on adeno 26, which is a human adeno. Gamaleya, which is the Russian Research Institute, which is actually visible through its international arm under the form of the RDIF, the Russian Direct Investment Fund, is actually using 2 doses. The adeno 26 is the first dose, and the adeno 5 is the second dose. CanSino Bio, which also developed, similarly to the Russian situation, the vaccine jointly with the Chinese military, is using adeno 5. Sinovac and Sinopharm, as I was mentioning before, use an inactivated virus. Actually, Sinopharm has two candidates based on inactivated viruses. So you see there are really 3 technologies at play in the front runners: a classical, good old technology with the inactivated virus, typically a 2-dose, if not 3-dose vaccine. You have the RNAs, which are 2-dose vaccines as well, and you got the adeno vectors, whether human or simian, and those are actually either single dose or 2-dose regimen. And you have some very interesting, runner-ups, so to say. And I want to signal only 3. One is Imperial College, which is working on a very interesting self-amplifying RNA, whereas the previous 2 are not self-amplifying. You got Novavax, which is a company which had the misfortune of failing the development of an RSV vaccine, but had a very promising influenza vaccine. So let's hope that their vaccine is successful, which is protein nanoparticles. And CureVac, which is another mRNA interesting vaccine, also coming up. So as you can see, a number of modern technologies have been at play, and in the earlier phase 1, I'd just like to signal Reitera, which is an Italian company which is working on the gorilla adeno. The gorilla adeno having the advantage of being paradoxically a very small adenovirus and very efficient in infecting human cells, and Medicago, which is working with GSK and Dynavax on VLPs, virus-like particles. So as you can see, overall, even without singling out some of those vaccines, as you can see, there's a huge field. Some companies being vaccine companies, some were in cancer immunotherapy and found quite opportune to jump on the coronavirus opportunity, and some being academic centers that have the possibility to accelerate the development of their technology in the current situation. Now, what are the key characteristics? And I'm not going to go through the entire table, but I'd like just to point a few things which actually drive the acceptability by the authorities. First is your vaccine really authorized or registered by what is called a stringent regulatory authority, or by the WHO, which of course is linked to the demonstrated clinical efficacy in phase 3 trials. Linked to this, there's a notion of availability, which is what is the production capacity? As you would assume, and I'll be happy to answer questions there within the, of course, the limits of confidentiality. As soon as you have positive phase 3 results, you have no idea of the number of friends you have in governments who wants doses. So your production capacity is below. Of course, affordability and therefore price plays a role, and of course the number of doses in the scheme do play a role, because if you deliver, say, 1 billion doses, but it's a 2-dose or a 3-dose scheme, you essentially immunize respectively 500 million or 333 million people. And lastly, and it's been a subject of intense discussion everywhere, the logistics are not simple. Even standard vaccines, they need refrigeration. So in countries where cold chain is a problem, the only option to bring the COVID-19 vaccine to the population will be to piggyback on the infrastructure that has been put in place for the existing program of immunization, the routine one. But when you have, vaccines that require deep freezing or I say simple freezing, all of a sudden you're in a situation where the last mile logistics are extremely problematic, and instead of bringing the vaccine to the people, you have to bring the people to the vaccine, and so you end up in very complex logistics. And from the programmatic perspective, the execution of the campaign is difficult. Now, I'd like to address in this presentation very briefly one concern, and I've been receiving that question myself by more or less politely by some people on the social networks. Whether people should have doubts about the development of the COVID-19 vaccine, and whether it was just too short, and whether therefore we should have doubts about the outcome. And I'd like to point to the fact that essentially those vaccines are the fruit of years of research and progress. And I'm going to discuss very briefly 4 main technologies that are actually pillars of modern vaccinology that have been leveraged in the preparation of those vaccines. The first one is reverse vaccinology. So what is reverse vaccinology? It's about identifying the vaccine antigens from the genome of the pathogen. As you know, China has been publishing the genetic sequence of the virus very early on. I think it was December 2019. Now, reverse vaccinology exists since the year 2000, okay, and it's been used very extensively. There's one licensed vaccine, which is the meningococcal B vaccine, which is based on 2 or 3 proteins, depending on the candidate of the vaccine, and it was actually designed entirely through the principle of reverse vaccinology. So first pillar of the vaccine science is not new. Okay, we were actually in 2020, we were essentially leveraging 20 years of pre-existing research. The second one is what is called structural vaccinology, which is about understanding the structure of the antigen so that you know what the antigen should look like in order to elicit the proper immune response. Here again, it's a science that originated in the early 2000s, and it gave a number of vaccines, such as the hepatitis B virus vaccine, such as the RSV vaccine candidates, and of course contributed to the design of the COVID-19 vaccine candidate. So here again, there's nothing new here. It's just a science which is reaching maturity, and it has been yielding fruit in the context of the current pandemic. The third one is so-called synthetic biology. So synthetic biology is the notion of using a synthetic gene for vaccination or for cancer therapy. Now, it was all started in 1986. Okay, mind you, in 1986, I was graduated from my from School of Pharmacy, but not much more. It was my first year in the vaccine industry. First position in the vaccine industry. And since then, as you can see from the historical chart, you have plenty of vaccines that have been developed from this on the basis of this technology by cloning genes that induce the production of certain proteins. So here again, nothing really new, and all the RNA and the viral vectors are all about understanding which genes encode which protein, and therefore making sure that the viral vector expresses on its surface the target spike protein. And lastly, there's the science of adjuvant, which is not really relevant for RNA vaccines or for the viral vectors, but very relevant to base vaccines in order to induce a broad and more comprehensive immune response. Now, the science of adjuvant was developed in the 1930s. Or so I say things in the 1920s, yes, rather. So it is not new, and we're essentially alumnus, and since then various adjuvants, essentially emulsions, have been used, quite extensively. And you have on the slide a list of the vaccines that have been leveraging those adjuvants. The last pandemic case was the pandemic influenza. So as you can see, adjuvant is certainly not a new science. So I guess the point I would like to try to drive home with those slides is that maybe the development of the coronavirus vaccines can look short, but it is actually leveraging, again, years, decades of vaccine science. And what you see is, why those technologies have been used, because the minute the internet provided the sequence, the synthetic gene was known, and then it took from anywhere from 1 week to 2 months to the various technologies. The oligonucleotides one are the fastest, viral vectors come next, and then protein expression purification comes last in getting into clinical trials. And that explains the speed at which those vaccines can be developed. Now, I think I'd like to say also that we have previous cases of fast development. The mumps vaccine in I think it was the late 60s or early 70s was developed in 4 years, and the Ebola vaccine, which is, as you know, all the candidates are based on the viral vectors, was developed in 2.5 to 3 years. So again, the point is, when someone is telling you that you cannot trust the vaccines because they've been developed too fast, I think these people don't understand the reality. Now, moving on, what are the benefits of those novel vaccine technologies in terms of improving acceptability? When you read, I'm not going to go through the entire matrix here. This is a classical matrix when you want to gauge assess the value of a vaccine candidate. You look at acceptability, availability, accessibility, affordability, and how to make the whole thing sustainable across all the stakeholders of the value chain. And let me focus on just a few things that are circled in red. What drives acceptability for the vaccines, especially in a pandemic situation, is the collective benefit seen from the perspective of the authorities. What do they want? They want the economy to get back on its feet, and they want to stop the problem. Whereas at the end user level, acceptability is driven by individual benefit and the perceived purity and the risk of the side effects. Those two notions are often hard to reconcile. Of course, availability comes into play. Do you have the right infrastructure, and can you reliably supply? I'm sure everybody has seen in the news, all the arguments going on regarding the speed at which the campaign can be rolled out depending on when doses are available. Access comes into play, whether you have something, a vaccine which is heat stable or not, and whether you have infrastructure to accommodate those or not. And overall, what makes the whole scheme sustainable and what actually ensures that you get to a sufficient level of immunity and people willing to re-immunize in case there's a need for booster shots is whether they have the right level of trust in vaccines, in addition to having the right amount of supply. Now, vaccine acceptability, I think it's fair to say, is a massive hurdle. This is a relatively recent data, and the bars are showing, in blue those who will a priori get vaccinated in the lay public versus those who will not. And you can see in countries like, to take France, which is very close to Switzerland, you could see that the vaccine skepticism is actually rampant, and it is the case also in some other European countries. Now, with that level of doubts, it makes you wonder what the authorities have to do to essentially push people, encourage people, if not coerce people to get immunized. Otherwise, with this kind of percentages, you will never get to herd immunity, and that is a concern from the perspective of the public health authorities. Now, the second concern, of course, is equitable access. This is a map which is, I think, for yesterday's or the day before yesterday, the UNICEF heat map of vaccine distribution. And as you can see, there's a massive imbalance in terms of number of doses relative to the population, as reflected by the color. And there are plenty of middle and low income countries who have not yet had access to the vaccine, and some of them may not see their first doses until the end of 2021 or the beginning of 2022, which raises, of course, a number of ethical questions. Now, very quickly, because I'm sure I would anticipate some questions. If I had the choice between all the technologies, what should I be getting? Well, I mean, the synthetic RNA vaccines are already on the market, and they're fast to develop. Among the unknowns is the safety, although this is improving quite a lot. Right now we're talking about millions of people immunized, and the rare side effects are known, and thank God they're very rare. And most of them are actually, transient and mild and easy to manage. We still don't know what is the long-term immunogenicity and efficacy. And of course, there are still some questions about acceptability. You have some people who believe that if they're injected with RNA, it would actually reprogram their DNA, and therefore people are afraid to be converted into some kind of horrible sci-fi like human GMO if they get this shot. For viral vectors, the strength is that we have several options. Time to market is relatively good. The unknown is the capacity, because they're a bit more complex to manufacture than the RNA. And the question is also, could you boost them? Because there's always the question of whether the subject would develop immunity against the vector itself, which could potentially, or at least theoretically, diminish the response in case of a booster. And lastly, you have protein vaccines, where there's a lot of experience. So this is very well-known technology, but of course, time to market is a bit more of a problem relative to the other products. Now, sorry, it's a bit of a busy slide, but it was one of the questions that surfaced when Salman and I were discussing the preparation of that presentation, which is, do we have a consensus on how to gauge the public health utility and the acceptability of the vaccine candidates? And I think now the consensus is yes, we should prevent severe infections rather than all infections, because essentially asymptomatic or mildly symptomatic infections are not a problem. Especially in the case of the new variants, you need to protect against the severe cases that maim or kill people and fill the intensive care units and have a huge impact on the economy. So this is why the key indicator is really not the number of cases, but it's rather hospitalization and fatalities. And so what is the kind of goal that is being pursued nowadays is to turn COVID into a mild illness, similar to the endemic coronaviruses. We will not be able to get that thing out, okay, completely. Everything indicates that we are going to migrate to a situation of endemicity with this vaccine. So the vaccine should prevent severe infections, and they will be accepted by the public as soon as there's a demonstrated safety profile. And as more and more people get immunized, you will have an increasing number of people accepting to take the jab. Of course, the clinical results could be a little bit more nuanced. There's a debate as to how you define clinical efficacy. You may have seen an interesting paper in the British Medical Journal, which was criticizing the way Pfizer and Moderna are claiming 90% plus efficacy. But the reality is what matters again is the efficacy against severe infections. And again, because this virus is going to become endemic and is essentially conveyed the same way as RSV, metapneumo, adeno, influenza, and so on and so forth, the idea is we will not be able to completely shield the population from the circulation. So the question is just to mitigate the morbidity and the mortality. And of course, we have to deal with the fake news and the misperceptions, because there are so many hair-raising stories out there that it creates a background noise, which is certainly not helping convince the vaccinators. Now, if you're asking me which vaccines should you choose, I think the most important sentence on this slide is the last one. Get the first one you can get, okay? Because essentially all the vaccines are commercialized surpass the 50% effectiveness level. All of them are effective against severe cases. The notion of waiting for the one that has the best numbers is like, waiting the next generation of mobile phone to buy a mobile phone. You never buy a mobile phone because every 6 months there's a new generation out. So of course, if you are high risk, if you have comorbidities, if you're a senior, by all means do get vaccinated, don't wait. And vaccinating in a respiratory disease, because the pathogen is airborne, this is the field, the type of viruses that spread the fastest. Thanks to modern life with international travel. Remember that the Spanish flu in 1918 killed anywhere between 50 and 100 million people at a time when people were traveling by train or by boat, and airplanes were actually small things that were not, transporting passengers. Okay, so just imagine if the Spanish flu was hitting today. And I can tell you this during the Q&A because this was part of the scenarios during the 2009 pandemic. So moving on, so of course, the vaccination strategy now from the perspective of the authorities is evolving. Why? Because we now have real life data. You may have seen the fantastic results coming from Israel with showing a very clear decline of the cases in the population. So all those results show that those people that have had COVID before, maybe one shot should be sufficient because the infection primed the immune system, and then what you have is and I see a question coming in the chat box. One shot is pretty sufficient for those who had the COVID-19, even those who had a mild case. So to the question asked by one of you, yes, I think going broad, doing one dose is probably preferable to going deep and making 2 doses, except in the frail and those with comorbidities. We know also that neutralizing antibodies from animal studies are now a good correlative protection, but we still have to wait a little bit to know what is the correlative protection in humans. So we should know very soon what is the magnitude of the immune response that's needed to protect, which is good news because when you think about it, for all the late runners, they will not need to run large-scale studies with 40, 50,000 subjects. They will do maybe a 5,000-6,000 subject immunogenicity study, and that would be sufficient. And the other thing is about the delay between the doses that I like to mention. It's right now those vaccines have been developed with a relatively narrow window of time between shots. Typically, it is better to space out the shots. This is what vaccine science is telling you. And actually, there's some thinking now being given to giving a third shot to boost the immune response, even if it's not specific to the new variants, to raise the level of foundational immunity, and therefore, have a lower incidence of moderate to severe cases. And increasingly, what you will see is also the try of what is called mix and match, meaning using different technologies, one for the prime, the second for the boost, because then you have, apology for the typo here, a more comprehensive immune response by actually boosting the immune system or triggering the immune cascade through different routes. Very briefly about equitable access, it's fair to say that there's an uneven distribution of where the vaccines are going, also because there's a certain imbalance of where the manufacturing units are, and this is a perennial question. What we have to hope is that ahead of the next pandemic, there will be more manufacturing sites that are ready to get started for pandemic management in the Americas, especially South America, in the Indian subcontinent, and in Southeast Asia. Now, are there any insights that we can draw from the current pandemic, and how can we better prepare for the next one? Because make no mistake, there will be a next one. The question is not whether we will have one. The question is when will we have one. And so you need to bear in mind so that the COVID-19 is not the 100-year event, which is what some people are claiming. Okay, 1819 in the early 20th century was a killer. Okay, remember also that the population of the planet at the time was about 25% of what it is today. Right, so just imagine in a world of 8 billion people, if something was spreading at the speed of EasyJet and Ryanair. Okay, just imagine what the death toll would be. And since 2003, it's not the first coronavirus. I mean, 5 of them have crossed from bats to humans, including SARS-CoV-1 and MERS. MERS is vectored also by the camels, but you need to know that MERS originated in Central Africa, where the herds of the camels are reared, and the baby camels are then brought to the Middle East through caravans, and this is how it actually arrived in the Middle East. And there are plenty of drivers of what is called zoonotic diseases, meaning diseases that jump from animals to humans. We travel. And we travel to countries where you have, live animals and bird markets. You also have cooking habits, or you could also have, the growth of the human population, which, results in encroaching on wildlife habitat and using wildlife as a food source. And you've got also farming practices. Every time we grow the size of farms, in Latin America or in the densely forested areas of Africa or Asia, you actually bring human beings closer to animal species that normally do not, I would say, live close to humans. And viruses are perfect weapons. They finally find a way to jump from an animal into another one, and we have to remember that we're mammals in this respect. So and therefore, for those reasons, it will only, the risk is there and will continue. So plenty of work is currently put together to prepare for the next pandemic. Mind you, some of those ideas were already on the table at the time of the H1N1 pandemic, starting by a better surveillance system, better international coordination, fast genetic sequencing. Remember, this was the single most important accelerator in the fight against the COVID-19 pandemic. You need to have access to animal facilities because, mind you, doing animal studies that have biosafety level 3 containment is not easy. And this has been a limiting factor for the companies that came in late, because they could not have access to CROs having BSL-3 animal facilities. You need to localize production units in the countries close to the population, where you have agile technologies such as RNA, such as viral vectors, such as protein expression system, and with the relevant amount of cold chain capacity next to you to store your product. And evidently, you need some funding, but this is relatively well addressed. And you need also public communication, because in the end, the more people resist the jab, the more you essentially delay the benefit of having the vaccine available. Just to illustrate this, CEPI, which is the Coalition for Epidemic Preparedness Innovations, has been created precisely for that matter. You have here a list of the viruses, of the diseases that they work on. And as you can see, some of them, Ebola is known. Marburg is a horrible thing. Lassa as well. MERS is known. And you can see there are plenty of disease. Some of them being vector-borne, so they're not spreading as fast as the airborne ones. And they always have a compartment which is called disease X, which is what if there's something completely unknown that surfaces. Mind you, the disease X compartment in 2019 was filled with SARS-CoV-2. And essentially, what will we evolve to? Several voices, and I'm citing only one, Stanley Plotkin. I have the highest respect for this gentleman. He's now in his late 80s or early 90s, and he's one of my masters during my life in vaccinology. And he says it very eloquently. He says that we are going to be evolving to an influenza-like model, where we'll need to have yearly or almost yearly changes of the vaccine, or maybe we'll need multivalent vaccine. And we'll need to have regional surveillance, we'll need to have agile technology, and we'll need to keep immunizing. Therefore, we'll need to have enough vaccine accessible to the people and at affordable prices, because the world will not be able to immunize against the coronavirus at large scale if the prices remain too high. Which takes me to the Q&A, and I'm sorry I've been a bit longer than planned, so let me stop sharing my screen, and I'm happy to take any questions that you may have.